everybody. So thank you very much all for joining us to listen to Georgina's talk. And thank you very much, Georgina, for, for coming and giving us this amazing talk. Okay, so before I introduce Georgina, I would like to I have a few housekeeping details to cover. So this talk is recorded, will be recorded, and we will mute you when Georgina is going to give her talk. And then when we have questions, so then you'll be able to ask her the questions. And if you can as well, please send a message on the chat when you need to ask questions. Okay, so I met Georgina at Highgate School, our local school, a few years ago when she gave a fantastic talk to the pupils and their parents. So, and she worked at UCL, where she set up the forensic DNA research facility, with whom she continues collaborations. So Georgina has also practiced as a forensic scientist working at the Forensic Institute in Glasgow, for which she continues to provide advice and consultancy casework. Now she lives in Australia, on the opposite side of the world, and it was really difficult to find a suitable time for her to give us this talk and on her latest research. She is at the moment a senior lecturer at the Centre for Forensic Science at UTS, University of Technology, Sydney. The main focus of her research is the transfer and persistence of DNA and how this affects the interpretation of DNA evidence in criminal casework. Thank you very much, Georgina, for giving us this very, very exciting talk. And I'd like now to hand over to Georgina for her to tell us more about the fascinating world of forensics.
All right, thank you so much, Paola. Well, as Paola mentioned, there was a time when I did work as full-time as a forensic scientist for a consultancy firm, the Forensic Institute in Glasgow. And it was there when I really learned a lot about DNA transfer and the issues within casework as a result of considering DNA transfer. And that's ultimately why I made the move into academia, so that I could conduct some of the research that's really needed to better understand DNA transfer and also to inform expert opinions that are given in court on DNA transfer. And this is something I'll talk in more detail as I go through today's presentation. Since being in Australia, I have continued on with the DNA transfer research here, and I've been very fortunate to be able to collaborate with partners at the Victoria Police in Melbourne and also at the New South Wales Police here in Sydney, to try and ensure that my research is actually informing everyday practice and helping improve the way in which we interpret and recover DNA within forensic science. So I'd like to start today's talk by giving a case example. It's quite an old case example now, and I apologize to Paola, who I know has seen a previous talk of mine and will be familiar with this case, but it really illustrates one of the key points I'm trying to make with today's talk. So this was the death of Mr. Ravish Kumar in California in November 2012. He was a billionaire at home in his mansion with his ex-wife at the time, when intruders broke in and with the intent of ransacking and stealing items from the house. In order to do that, they tied up Mr. Kumar and his ex-wife, and they used duct tape to do that. And they put duct tape across their mouth and nose to obviously keep them silent, and went about the home invasion. At some point during this, the ex-wife was able to get free and call for help. Unfortunately, by the time police and paramedics arrived, they found that Mr. Kumar had suffocated to death. So later, his body was taken and examined as part of this criminal investigation, and samples were taken from his fingernails. These were examined for DNA, and a mixture of DNA was found. Some DNA coming from Mr. Kumar, as you might expect, as they were his fingernails, but also another DNA profile. this was identified to have been coming from Lucas Anderson. And Lucas Anderson was arrested and charged with this crime. However, what came to light later was that at the time of the home invasion, Mr. Anderson was actually in hospital, unconscious, and with a blood alcohol level of 5 times the legal limit for driving. So my question to you is, would you continue on to charge and convict Mr. Anderson based on the DNA evidence? Please feel free to put your thoughts in the chat. Okay, I think maybe you're all just a little shocked perhaps by this case, and quite rightly so. The question, of course, comes is, how did Mr. Anderson's DNA, assuming that the DNA has been processed properly and interpreted correctly as likely to be coming from him rather than someone other and unrelated to him, how did his DNA get onto the fingernails of Mr. Coomer? And it was up to the defense team, or his defense team, to investigate this and try and identify. And what they discovered, that when Lucas Anderson earlier in the day had collapsed on the sidewalk due to his intoxication, paramedics had attended him and taken him to hospital. And then later, when paramedics went to the scene of Mr. Coomer's death, it was the same paramedics that attended him. So what they think is that this little device that you clip onto the end of a finger to measure oxygen saturation of the blood, that is the culprit that transferred Mr. Anderson's DNA to Mr. Coomer. So this, as I mentioned at the start, it gives a really clear illustration of why we need to consider this concept of indirect DNA transfer. This idea that your DNA can go places and end up on items where you've never been and that you've never touched. So what exactly is this indirect DNA transfer? Well, it was first identified in a key Nature paper published in 1997 from a researcher and casework scientist at Victoria Police. And what they showed is that when you touched an item that someone else had already handled, you could transfer their DNA onwards. And also when you shake hands with someone, you can transfer their DNA onto an item. And that led the way for quite a few research studies into the early 2000s that essentially repeatedly showed that if we clean an item that's DNA free, like a beaker in a laboratory, ask volunteers to shake hands and then handle the item, when we recover DNA from the item, we can get a variety of results. We might just get DNA from the person who touched the item, but we might also get mixtures of DNA. Some DNA is coming from the person touching the item, and some DNA is coming from the person who shook hands. And then there's even some examples where you only see DNA coming from the person who shook hands rather than the person who actually touched the item. And essentially a variety of results in between. So this essentially started the progression in research to try and understand under what circumstances does indirect DNA transfer happen, and when is it important to consider in casework. It also started the field of just general investigation into the variables that impact direct DNA transfer, that is the transfer of DNA from your hand, for example, if you're using your hand to touch something, directly onto that item. And there's a variety of variables that have been identified over the years that impact that. And I'm going to give you some illustrations of those to really emphasize the complex nature of this idea of DNA transfer. So one of the key factors that we often talk about, and that has been debated a lot in the literature over the last 20 years or so, with much more investigation in the last sort of 5 years or so, and that's this idea of shedder status. This is where we know that the amount of DNA you shed or deposit onto an item when you touch it can vary between individuals. And there's this really nice study from a group at Flinders University who illustrate using a fluorescent dye that binds to DNA, the difference in the amounts of DNA deposited by people with different sheddar statuses. And we're generally categorized into 3 sheddar statuses: heavy or good, and here we see a distribution of DNA that's being deposited onto an item. Then intermediate, so we're seeing a bit less DNA being deposited on an item. And then light, so even less DNA being deposited on the item. And as I say, essentially we can categorize people into these kind of 3 categories, but more recent research is starting to show that it's not quite as clear cut as that. And it can depend on a lot of variables, such as the time of day, what someone's done immediately before handling something, and other factors such as the condition of someone's skin can impact how much DNA they leave. We know that people with skin conditions such as eczema, they're likely to leave more DNA than someone who doesn't have a skin condition. And that kind of makes sense logically when you think about the flakiness of skin that can be associated with these sorts of conditions. We know that the excuse me. We know that the age of the donor can impact how much DNA they leave too. So for example, a couple of studies have shown that DNA deposition tends to decrease as you get older, and this is particularly observed for men rather than women. And then I mentioned this idea of like activities prior to touching something, and this can impact how much DNA you have available. And it can impact how much DNA you have available in 2 ways. One is that you might collect up other people's DNA, and we know from a variety of research studies that up to about 15% of DNA on your hand is actually coming from other people. And then that's commonly what we refer to as close associates. So that could be people that you're living with, it could be a romantic partner, it could be your children, but also just other unknown DNA that you're kind of picking up as you go around the world in your everyday life. So for example, here we see someone handling a rail on the street. That rail could be handled by many other different people, and therefore they could pick up DNA and move it on elsewhere. We can, and other parts of our body, we can load up our hands with our own DNA. So depending on what we've done immediately before touching something, this can really affect how much DNA we leave. And then the type of surface that we're contacting can have an impact, whether it's porous or non-porous. And then the type of contact that we make, how long we're making that contact, how frequent is it, is there friction involved, is there pressure? And we published a study from one of my master's students at UCL a few years ago that showed that when you put more pressure onto an item, you do leave more DNA. Again, sounds quite obvious, but as scientists, we want to make sure that any opinions we provide are actually supported by data. And this was the first study that actually demonstrated that. And then there are many other factors, some of which we know and some of which we don't know because we're still really investigating this. So if we move forward then from direct transfer, hopefully I've now illustrated to you quite how complex it is and the variety of variables that we might consider when we're thinking about how much DNA do you leave when you touch something. If we then go on to think about indirect transfer, well, first of all, this is very much dependent on how much DNA was initially deposited that's then available for that onward transfer. And of course, the amount of DNA that's initially deposited is completely dependent on all of those variables I've just mentioned. Furthermore, the type or the biological source of the DNA that's being deposited, for example, if it's a body fluid like blood or semen, or if it's just trace DNA that's being deposited through touch, that can impact how readily it will transfer on, and also whether that biological material is wet or dry. And then finally, we know that there can be a lot of steps involved in indirect transfer. What I mentioned earlier about this idea of shaking hands and then touching something, that's referred to as secondary transfer. It just has one intermediate step between the individual and the DNA, the surface on which the DNA ends up. However, there might be tertiary transfer or quaternary transfer. So with each transfer step, less DNA gets transferred. So this also impacts how much DNA we might expect to find at the end. And then, as with direct transfer, there are still going to be many more variables that we don't yet know about and we need to identify. So now that I've talked about different variables and how essentially complex this concept of DNA transfer is, I'd like to talk about when does it actually occur when we think about a crime and when we're thinking about the investigation of a crime scene and ultimately giving evidence in court on the DNA findings. What do we need to consider? Well, first of all, before a crime even occurs, any location could ultimately be a crime scene. And of course, we don't live in a clean world. So there is background DNA everywhere, essentially. Just to give you some examples of that research that I've been investigating this level of background DNA, Reither et al. Showed they investigated background levels of DNA on flooring in residence in people's homes. And they looked at living rooms and kitchens and bathrooms. And they found that on average, they would recover DNA samples that contained at least 3 individuals of DNA. And more than that, what they observed after cleaning the floor was that there was next to no difference in the DNA and the number of contributors that were identified before cleaning and after cleaning, which just shows the kind of prevalence of background DNA in our lives. Similarly, we've done some research, again at UCL with a master's student a few years ago that we're just writing up for publication now, where the student looked at recovering DNA in a communal kitchen area within an office building. And again, we saw an average sort of 2 to 4 contributors being present within the mixtures of DNA that were detected depending on the nature of the surfaces that were being sampled. And then finally, there's been quite a lot of research in more recent years starting to look at regularly used items and clothing. And we see that there are always background levels of DNA. It can be all the way up to several nanograms of DNA, which is actually quite a lot. So we have this knowledge now that even before a crime occurred, any kind of location, whether it's indoor or outdoor, there's going to be an element of DNA that's already present, and that could be recovered as part of the investigation of the crime scene. And then, of course, DNA transfer will happen during the crime. So let's say we're dealing with a crime against a person, and we have one offender and we have one victim. So we expect that there's going to be DNA transfer from the offender to the victim, but also from the victim to the offender. So it's bi-directional. We would expect to see DNA transfer from the offender to the scene. And by the scene, I mean surfaces within the crime scene, for example, the wall, the floor, the door, but also items within the crime scene. Similarly, we'll have transfer from the victim to the scene, but remember it's bi-directional. So they're going to be picking up DNA from the scene as well. That background DNA that I've already mentioned. And then remember, we're not just talking about direct DNA transfer, but also indirect DNA transfer. I mentioned this idea that we are carrying, up to 15% of the DNA on our hands coming from other people. Well, that DNA we're bringing with us and we're taking to environments. So at a scene, an offender and a victim are going to be bringing other people's DNA with them. And of course, that might ultimately get recovered during the crime scene investigation. And then when a crime occurs, it's not always instant that it gets reported and the scene gets secured by the police. There can be a delay between the crime occurring and the crime scene being secured. So during that time, there might be some DNA transfer events occurring. For example, the offender might try to clean up. I've certainly worked a case back in the day where an offender did try to use bleach to clean down the crime scene. We know that offenders might even just take a cloth and try and rub down surfaces, ostensibly trying to get rid of finger marks. But we've also done some research that suggests that actually just the physical rubbing of an item will actually remove quite a lot of the DNA. But also other groups have observed that physical rubbing with a cloth, for example, can then also transfer DNA onto another surface because that cloth itself acts as a vector. Depending on the nature of what becomes a crime scene and how long that delay is for, there might be people going about their routine activities within that area, or there might be animals that disrupt and move items and could move DNA about. And again, we're having both direct and indirect DNA transfer going on. And then finally, we expect to see, or DNA transfer can occur during the actual crime scene investigation and the laboratory examination of any items recovered. What research tells us is that when you attend a crime scene and some of the activities that perform within a crime scene, for example, kneeling and over an item and speaking will deposit DNA, certain activities like beckoning over your colleagues or things that you're doing like routinely will potentially deposit DNA. We also have this idea from, typical crime scene shows that gloves are the solution to everything. We'll just put on a pair of gloves and we're not going to contaminate the scene. However, more recent research is showing that actually gloves straight out of the box aren't always DNA free. They can already have DNA on them just as part of the manufacturing process because they're not manufactured to be DNA free. Similarly, using gloves, using tools, even using fingerprinting brushes can all transfer DNA from one item to another or from one surface to another. So this means that we need to be aware of these things and therefore try and mitigate it. So what can we do? What does this knowledge about DNA transfer, how can we use that to improve our practice within casework? Oh, and sorry, one final point I forgot to mention is that further to the tools themselves acting as vectors of DNA transfer, if we package an item at a crime scene, let's say like an item of clothing, ostensibly we're packaging it to preserve the evidence and then sending off that item to the laboratory to be examined. But what research has shown is that DNA can transfer from the item into the surface of the inside of the bag, but also can redistribute to different locations on the item. So let's say you're examining that item of clothing, let's say it's a jumper, it comes from a victim that is saying that they're a victim of an assault, perhaps they've been grabbed on the jumper. You might be then targeting particular areas, say the front of the jumper, for DNA that could be coming from the offender. But in the way that that's been packaged, if DNA has been moved around, DNA might have originally been in a different location, have moved to that location, therefore becoming more incriminating than perhaps it would have been otherwise. And these are things that we need to consider. So how do we deal with that DNA transfer in casework? Well, let's start with that area where we have the most control over, that area of the crime scene investigation and the laboratory examination. Well, firstly, we want to use personal protective equipment, wear a suit, wear a face mask, wear gloves. But more than that, we need to ensure we put on our PPE in the right order so that we minimize our transfer of our own DNA onto the PPE that we're wearing. We want to make sure we're wearing it properly. So for example, if we're wearing gloves and we're wearing a crime scene suit, one of my pet hates is that gap between the glove and the sleeve. And you see that all the time. You see that on both fiction shows, but you also see it in real life reporting in the media, that there's that gap. And really there shouldn't be, because that's exposing your skin and that exposes you to potentially depositing your DNA at the scene. So what you need to do is apply a first pair of gloves, tape it to that sleeve, and then apply a second pair, and then change that second pair in between handling the different items so that your gloves don't become a vector for DNA transfer. And then when it comes to packaging, we're currently doing some research at La Trobe University for a master's student there and an honours student here at UTS to try and better understand when DNA transfers within packaging and how can we package better to better preserve the DNA evidence. Something that's really important for forensic scientists and crime scene investigators to do is to ensure that they're following standard operating procedures. And laboratories across the world will have validated and accredited standing operating procedures, and it's really important for scientists to adhere to them. I think many jurisdictions across the world, including the UK, have had high-profile cases where there's been cross-contamination from one case to another of DNA, when someone's then resulted in someone being wrongly charged for a crime that they obviously weren't involved in, but because someone didn't follow a standing operating procedure that led to that cross-contamination. But the thing is, of course, we're human, right? We are going to make mistakes occasionally. So what we need to also make sure is that we have a good audit of processes. And this can be internal through peer review within a particular case with laboratory, but it can also be the role of someone like myself who is instructed by defense lawyers and provides that kind of consultancy from the defense side of things, who can then review over the casework and the case files and ensure that the process of DNA analysis and recovery and analysis has been done to the appropriate standing operating procedures, and that there's been no opportunity for DNA contamination to occur from the moment an item was received at the laboratory to that DNA evidence actually being provided, being interpreted and provided as an opinion to court. But what about these other three areas that are not in our control? These areas that are happening before we actually secure a crime scene. How do we deal with DNA transfer in those elements of casework? Well, really, there is only one way in which we can do that, because we don't actually know what happened during any of these things, or in most of the time we don't. So what we need to do is we need to consider, what is the meaning of the DNA evidence? What is the likelihood of seeing that DNA evidence when we consider different activities that may have resulted in that DNA evidence? And usually we focus predominantly on two activities. One activity that the prosecution is saying happened, so usually that's the criminal event, and an opposing activity that the defense are saying that explains how their client's DNA got onto the item of interest. So what we want to do is determine what's the chance, what's the likelihood of seeing that particular DNA evidence if what the prosecution said happened is true, versus what is the probability, the likelihood of seeing the DNA evidence if what the defense is true. Now, when I worked in casework routinely over 10 years ago in the UK, you would quite often then see what I would refer to as one-sided reports. This is when the prosecution would say that the DNA evidence is as they would expect if this particular activity, the crime event, had happened. And most of the time that is true. That is what you would expect of the crime event. If it wasn't, you probably wouldn't find the DNA evidence in court, right? But the challenge comes is that, but would you also expect to see that DNA evidence if what the defense said is true? And that's something that always needs to be considered. You always need to have that alternative in order to provide a fair assessment of the evidence and ensure that the jury is presented with the right and informed information to make a proper and informed decision on whether someone is guilty or innocent. So then how do we go about informing those probabilities? And that's a really hard question to answer. Now, over the last few years, there's been quite a lot of debate within the forensic science community about how we do this. And a couple of years ago, the International Society of Forensic Genetics published their latest guideline on how to evaluate, how to interpret the DNA evidence, given these activities, either what the prosecution has said or what the defense has said. And what they said, what they recommended, was that the expert opinion must be underpinned by experimental data. Wherever possible, published data should be used. And so you will have seen I've referenced a lot of research studies as I've gone through my talk. And it's those kinds of studies that we need to be looking to when we start thinking about what does the DNA evidence actually mean when we find it at a crime scene. In order to assess situations like the one I gave, the example I gave at the beginning, with the case of the death of Ravish Kumar. Now, the kind of data that we actually need kind of falls into two categories. And therefore, what we really need is two different experimental study sets. And again, over the last couple of years, we've been publishing on this to try and help direct researchers to improve the quality of their research. So firstly, we need an experimental design that's going to help us better understand DNA transfer. When does it happen? And what are the impacting variables? And clearly, like any other form of scientific experimentation, these require highly controlled studies so that you can really isolate that variable of interest to know whether that's having an impact, and if so, by how much. And we also need repetitions. So we need the same volunteer doing the particular activity multiple times to get an understanding of what's the variation for one volunteer, and then looking at another volunteer and making that comparison. And in this way, we're just trying to better understand DNA transfer. And this is the kinds of studies that I've talked about mostly today. However, we then need to go on, and we need to actually generate the data required to inform those probabilities that I mentioned. The probability of seeing a particular DNA outcome given a particular scenario or activity. And these studies need to be more realistic. They need to represent case scenarios. And here, what we need instead of repetitions is we really need iterations. We need multiple volunteers doing the same action. So let's take, for example, if we're thinking about a stabbing case, we've recovered a mixed profile from a knife handle. We want to know, what's the probability of seeing a particular DNA profile if it's come from the person who actually handled the item, handled the knife, did the stabbing. All right. So let's say we did an experiment where we asked 100 volunteers to take a knife, stab it into a phone block. And then we looked and we recovered the DNA from the handles, and we looked to see how often did we see DNA just coming from the person who handled the knife and stabbed it? How often did we see DNA coming from other people? What is the different proportions that we see? And in this way, we'd essentially build up frequency database of better understanding that if we see a DNA profile coming from a knife handle, we'd start to be able to inform what's the probability of actually seeing that if that DNA did come from the person who actually handled the knife and stabbed it. The challenge that we actually have in the current research landscape is that we haven't done either of these things all that well. And I'm being critical of myself and my research and my group's research too, I hasten to add. I mean, we've done the first looking at those impacting variables, investigating DNA transfer pretty well. But what we've tended to do, and like I said, I'm guilty of this too, is that we've tended to try and create a study that does everything, that tells us more information about a particular DNA transfer variable, but also tries to generate data that's that's useful for informing probabilities for court. But what's actually happened is we've increased our understanding of DNA transfer, but we haven't really hit that sweet spot with generating the data for probability. And so right now there's kind of that gap at the moment that we really need to try and fill through research to actually provide casework scientists with the data they need to provide a robust, balanced and informed data-based opinion to court, again, to ensure that the jury is making a fully informed decision. When we talk about DNA profiling within forensic science, you'll most likely be thinking about, can we match a DNA profile that we've recovered from a crime scene to an individual? That's one part of DNA and the use of DNA, and one part of DNA interpretation within a case. However, this area that I've been talking about today is an area when we're talking about the context of the DNA evidence. It's assuming that the DNA is coming from the person that we believe it is, it's then taking it on to the next level of talking about how did the DNA get there? What's the likelihood of seeing that DNA given the different activities? And arguably, this is a different kind of expertise. And there are some jurisdictions around the world, for example, in the Netherlands, who are very good at recognizing this as a different area of expertise, and in fact have a process in place to assess their experts who go to court to ensure that they have the right level of expertise to make these kinds of opinions or provide these kinds of opinions on these kinds of issues of thinking about how the DNA got there. And I've been really fortunate to be one of the scientific assessors within that process to ensure that the experts they have going to court on this matter are indeed have the right expertise to provide this kind of opinion. All right, so really that leaves me just to say one thing for you as take home. I mentioned prior to actually starting the talk proper that one of the reasons I like giving public talks is that ultimately you all might one day act on a jury, and you might one day hear about DNA evidence. And so what I want you to know, and why I've sort of dwelled quite a lot on the sort of complexities of DNA transfer, is that it's not as straightforward as the DNA was found on the knife handle, therefore this person stabbed this other person. It's so much more complicated than that. You need to be asking the right questions. So what I want you to take home from this is that DNA does not automatically mean guilty. We need to actually think about what does the DNA mean within the context of the crime. Okay, thank you very much for your attention.
Very interesting, thank you.
I've just seen a question on the chat. How likely is it that tampering with DNA obliterates evidence on a crime scene? Do you mean like cleaning up the scene? That's where you were going with that?
May I speak?
Yes,
Yes,
Alessandra, yes.
Thank you. Yes, I'm referring particularly to voluntary destroying DNA evidence that has been dropped. And I refer particularly to cases of so-called bent officers who maybe know how to do it and
oh, okay.
Yeah, and deliberately destroy some evidence. I'm thinking of a case in Italy that happened many years ago, but it could happen everywhere. Thank you. And thank you very much for your talk. Incredibly interesting.
Oh, you're welcome. Thank you. No, I mean, people can. And it's kind of the challenge that you have really when you do this kind of research to see, hey, what cleaning methods destroy DNA? And sometimes people say, well, you shouldn't publish that because you're giving other people ideas. But at the same time, we need to understand this so that when we do get a DNA profile, say that it's, you know, what we refer to as a partial profile, it's degraded. Do we then have some idea of whether there's been some interference at the scene? Can we use that to help, you know, infer that or not? And there's still quite a lot. There's only been a few papers in recent years that started looking at cleaning methods. It's kind of similar to a lot of research that was done within fingerprints. And so, of course, there was a sort of transition that some offenders would start wearing gloves to crime scenes because they realized that if they were barehanded, they would leave, you know, finger marks at the scene. And so in some ways, it feels a little bit like an arms race, because as we learn more and we publish and we put it out there, of course, offenders are going to learn more. And of course, as you say, if people were to work within the environment and choose to then offend, they might well be a little bit more informed as to how best to go about getting rid of their DNA, of their finger marks and traces that they're leaving at the scene. So yes, it is possible for someone with knowledge of the subject to do quite well, I should think, of getting rid of their traces. That said, though, DNA really does get everywhere. So I would challenge someone in the stressful moment of committing a crime whether you'd actually think of everything. But I don't know, I'm not an offender, so you know.
So if it is claimed that no DNA evidence was found on the site, on the crime scene, is not really possible, is it?
I think it would be unlikely.
Hmm.
But I don't really know. I mean, like, it depends on time as well, right? If they were not discovered and they had the time to really bleach down the scene super thoroughly with a high concentration of bleach, yeah, maybe. I think it would be very strange for someone to attend a scene and then not get any DNA traces. I mean, especially as I was saying, like, you know, we don't live in a clean environment. There's DNA everywhere. There's an expectation that there's going to be DNA being recovered. Question is whether it's coming from the offender or not is more challenging. I mean, today I talked about transfer. I did sort of a little deep dive into transfer, but it's actually part of a bigger series of considerations that we actually refer to as TPPR, and it's transfer, persistence, prevalence, and recovery. And so it's thinking about how long does DNA persist for, how often, how much is DNA prevalent in the world, and what's the best way to recover it? Because also how we recover it will impact how much DNA we see as well. Okay, Paolo, do you want to read your question? Go for it.
Oh, yes. I was thinking how far DNA is traveling when we are coughing and talking at a crime scene. So yes, and thank you very much, really, Georgina, for giving us this amazing talk. Okay.
So I mentioned a study earlier in the talk, actually, from Guy Ratti and others, and they did exactly that. They asked people without masks to, when they were kneeling, they would talk, and they would cough, and they would see how far the DNA went, and they observed it. They were taking samples up to about, I think it was about 1.5 meters, don't quite quote me, but something in that region. And that's how far the DNA was transferring. Once they put a mask on, obviously that reduced that significantly. But as I mentioned, obviously wearing a mask, I don't know if I mentioned this or not, but wearing a mask can move if you're talking. So it can like slough off your cells here. So you can then still leave DNA, but not as far, but you might still leave DNA at the bottom. So if you're quite close to an item, you probably shouldn't speak over it even when you're wearing a mask, and then leave talking to your colleagues at the scene in a more general way rather than actually facing an item.
Yeah. And when the police come after a crime, so they could contaminate as well, isn't it? They don't always, it's not the first people who come, the forensic scientists, isn't it? The first the police come. Yeah. So
the police are first responders, absolutely.
Yeah. And how are they trained to avoid DNA contamination or something like that?
I hope so.
Yes. Okay.
The other challenge actually is paramedics. For absolute obvious reasons, paramedics' priority is life-saving attempts, and in fact, life-saving is normally the priority of any activities of first responders at a crime scene. And so, of course, paramedics can also contaminate a scene, not intentionally, but that can happen, and it's those kinds of things that what we ideally need, and I know that what the forensic science regulator is pushing for within England and Wales, is that we have what we refer to as elimination databases. So people who do attend scenes, like first responders, police officers, can provide their DNA so that the first sort of point of call when you're examining or when you're interpreting DNA that's been recovered from a crime scene is to check, has it come from a first responder? Has it come from a lab member of staff? So you do that kind of first check first to ensure that you can rule out contamination before then taking it on and comparing it against suspects or uploading it to the database or doing what you need to do with it.
Okay,
I had a couple of questions, Paola, that I typed in the chat. I don't know if it is possible, but I was wondering, thank you, Georgina, first of all, to tell us all these things. And now I will see all these things, films and movies with a different eye on it.
Yeah, you'll start criticizing them now.
I start criticizing from a PPI. And I was wondering, why don't you get to the point to get the DNA of all population, or do you actually have so much from all population, or just from people who already committed crimes? How do you compare things? Where do you get your models?
Okay, so many countries across the world and jurisdictions will have DNA databases of people who've been convicted of crimes. So that usually is the database that I refer to when I say you might upload a DNA profile to see if you can identify a potential suspect. And then some jurisdictions will also have what we refer to as these elimination databases, people involved in the forensic process who can be eliminated. In terms of whether you should have a DNA database of an entire population, personally, I would not want that. Because of these issues of DNA transfer that I've talked about today, I would be afraid that my DNA might end up somewhere it shouldn't be, and I could be accidentally implicated in something. And the reason I talk about the Lucas Anderson case is because he had an ironclad alibi, so it's really clear to illustrate that he definitely did not do that crime. He couldn't have. Whereas in everyday world, it may be just a case of saying, well, I didn't do it, I was asleep at home alone. And if you've got no way of sort of proving that, how do you? And I feel like there's quite a lot of onus now on the defense to actually provide an explanation for DNA. But if you were genuinely innocent, you might not know how your DNA got there, and I think that's quite challenging. And so for me personally, I wouldn't want my DNA on like a population database. That's my personal view.
Sure. Thank you, Georgina. Thank you.
That's all right.
I had other clarifications, but I don't want to keep you too long. It's okay.
Oh, I don't mind if you want to ask away.
Well, I was fascinated by what you mentioned about repetition. You need repetitions for, can't you just amplify the thing? Isn't there any technique, instead of having a repetition of a thing? You know what I mean? But yeah, the amplification.
So the amplification process is for making a lot of copies of DNA. So that's part of the process that we do during DNA profiling. You essentially make lots of copies of your DNA samples so that you can target particular areas and generate a DNA profile. When I talked about repetitions, what I meant was different activities, or the same activity being done multiple times by an individual, so that you can get an idea of variation that's happening just for one individual doing one thing. Does that make sense?
Yes, of course, yes.
Because like I talked about, we know that you can deposit different amounts of DNA at different times of the day, and depending on what you've done before and all those kinds of things. So we try and get an idea of what variation do you have just for one person doing something.
Thank you very much, Georgina.
May I ask another question?
Mm-hmm.
You did a talk in a school. This is what What may I ask what it was about? What was of interest in a school about DNA in forensic sense?
So for me, when back in London, I actually did quite a few talks at various different schools, and part of it is about wanting to be sort of a female mentor. The idea that women can go on and be scientists. So part of it is kind of promoting that. And part of it is the idea that science, I think science can have a poor reputation in a very general sense, right? It can be depicted as quite boring and also, like I say, male dominated. And so I think that going and giving talks about like what forensic science is, and here's an application of science that actually has a purpose and that can be useful and it can be interesting. And so the kinds of talks I do for them are a little bit different. Obviously today I've really focused a lot on the research and the use of DNA, but for school it's more about the process of DNA, and also what does a forensic scientist look like? What is a role of a forensic scientist? And we also get bombarded with a lot of images, in the media, and you see that it's the same person that goes to the crime scene, who then does the analysis in the lab, who then goes to court. But in reality, that actually doesn't happen. And so it's also about ensuring that students at school have the fully informed perspective of what does a forensic science role look like. There are different parts to the process. it'll be one person at the crime scene, it'll be a different person examining an item, recovering, say, for example, biological materials from an item. There'll be a different person processing it for DNA profiling in the lab, and then it'll be a different person interpreting all of that and taking it to court. So yeah, if people are, youngsters are thinking about what do they want to do in the future, it's important for them to actually have that information to make informed decisions of what their future will look like and what do they want to work toward. I'm sorry, did that actually answer your question? Yeah, okay.
Okay, yes. Thank you, Georgina. Thank you. And yes, I have another one. What about identical twins? Can you tell them apart because they have the same DNA, do they?
Yes and no. So with routine DNA profiling, the way in which routine DNA profiling works is that you just target specific areas you know differ between individuals. And those areas are made up of repeating sequences of DNA. And we look at the number of repeats. So that means that between, as you say, between identical twins with identical DNA, those number of repeats are going to be the same. So with a routine DNA profile, no, we can't tell them apart unless one of them accidentally had a mutation where the other didn't, something like that. Generally speaking, no. However, there is more that we can now do with more advanced technology. One of the things we can now do is sequence the entire genome. And there may well be some single letter differences in the code of the DNA, potentially, that could be identified. And the other way is what's referred to as methylation. So it's a chemical addition to the DNA molecule that occurs at various different points throughout our lives. So within the womb, the environment within the womb, this kind of chemical modification happens. It doesn't affect the genetic code. It's just like literally just these chemical additions to the DNA structure. And then throughout our lives, our diets will impact how much of this kind of chemical modification that happens, whether we smoke. Trying to think, there's various other things that modify these chemical additions to the DNA structure. And so that's called methylation. And so we can actually see different patterns of methylation between identical twins just because they've been exposed to different things in their actual, adult lives, essentially. So that is one way that we can distinguish between identical twins.
So is the DNA of, for example, the skin or the blood or have different methylations, or will it be the same?
So yeah, different body fluids can have different methylation patterns, and that's something that is currently under research to use as a way of identifying body fluids. Because one of the challenges that we have now is because our DNA technology is so sensitive, when we recover DNA, let's say from a dilute blood stain, you can't be really all that confident necessarily that the DNA is coming from the blood. It could be coming from the surface that was underneath blood. Or it could have been deposited like, trace DNA after the blood's been deposited. And so what sort of one of the, I suppose, holy grails of forensic biology at the moment is to try and develop a test where you can actually associate the DNA profile with the body fluid. And methylation is quite promising in that because it means, because the methylation is happening on the actual DNA that you're also looking at, if that makes sense. So yeah, there's quite a few things, research avenues looking at the detection of body fluid.
Yeah, this is so interesting, isn't it? So many details. Okay, so yeah, is anybody else would like to ask any questions? Doesn't seem to. Okay, so yes, so I think we it was really so lovely. It's a pleasure to listen to your talk, Georgina. And yes, maybe I pass the I would say to Simona. For Simona, would you like to say something?
Yes, just I would like to thank Georgina. It was really interesting and very different from our other talk that we had in the past. So thank you very much. Thank you for your time. And I know it's quite late down there in Australia. Thank you.
Oh, you're very welcome, and thank you for inviting me to speak.
Thanks. And it will be on our YouTube channel next week. So,
Okay, great. Will you send me the link? And then I
of
can...
course, yes. We will.
Yes, yes,
Okay.
of course, yes.
I know my
yes.
parents always like to watch these things.
Good.
Oh,
good.
lovely. Yes, so that's great. Thank you very much, Georgina.
All right,
And,
thank you.
Okay, so have a nice evening.
Thank you. Have a good day.
Yes, yes, thank you. Yes, it's really strange, isn't it? You're finishing the day and we are starting the day.
I know, exactly.
Yes. All right, okay. It was really so nice to see you and yes, to catch up with you as well. All right, thank you.
Thank you.
Thanks so much.
Thank you.
Bye-bye.
Bye. Bye.
Bye.